All right, Monica, now the floor is yours, I promise.
Awesome. Thank you, Michelle. Thanks for the introduction, for the invitation. And, I'm happy to be back, virtually. Much as I love, Eastern Washington in the winter, I'm not complaining about not having to travel today. So, I will kick it off and talk about our experiences with grapevine leafroll disease management, specifically some of the challenges and then opportunities that I see that we had and that you as growers in Eastern Washington also have. So welcome and thanks everyone for joining us. So just to get a little bit of the basics at the start, leaf roll disease, there are 6 viruses that are associated with leaf roll disease. So the grapevine leaf roll viruses. This is an incurable disease. So once the virus has infected the grapevine, there's no way at this point that we can cure the vines from that infection. So that means that the only way to cure them or the only way to get rid of the inoculum is actually to remove those infected vines from the field. And that's something that we'll be talking about. And then the reason why we're talking about this today is because leaf roll disease has effects both at the, sort of general level of the vine on vine health overall, but then also specifically on ripening of the fruit and anthocyanin production, sugar accumulation. And if you have a chronic leaf roll infection, you do see reduced yields. And as I said, effects on plant health, mainly associated with the fact that the vine is just not photosynthesizing properly. And then also because these are phloem limited viruses, even the photosynthase that are produced, the nutrients don't move around very well in the vine. So there are those impacts that you see on vine health. Of the leaf roll associated viruses, the grapevine leaf roll virus 3 is the most common worldwide. So when I talk about leaf roll virus, I'm really going to be talking, and management, I'm really going to be talking about leaf roll virus 3. On the vector side, as Michelle said, this is a pathogen that is moved around by an insect. For some reason, I seem to gravitate towards those, maybe because they're extra challenging to work with. I'm not really sure, but it definitely keeps us busy. And so for leaf roll virus 3, the vectors are mealybugs and soft scale. In California, we have 5, at last count, we have 5 different mealybugs in vineyards. The 2 most common are vine mealybug and grape mealybug. And then we also have soft scale. So we have European fruit lecanium scale, which is also a vector. However, as far as, this virus has been studied worldwide, most of the studies have focused around mealybugs as vectors. And since those are our main vectors, those are the only vectors that I'm going to talk about today, other than to mention that they are not the only vectors. So in Washington, unlike in California, you have one mealybug species in vineyards, and that's grape mealybug. So that is a principal vector of leaf roll 3 in Washington. It's an insect that's native to North America. So what that means for us is that it's a somewhat sporadic pest. So it may be very widely distributed, but it doesn't often reach large populations to become a direct pest itself. So the reason why we're so concerned about grape mealybug usually is because it's a vector of these viruses. So not because it's causing economic damage on its own. Occasionally you get large populations that are doing that, but generally just because it is moving this virus and then the impacts that the virus have on vine health and fruit quality. So it can actually be kind of hard to find grape mealybugs. So we, at certain times of the year, we'll cut apart the clusters, looking for the grape mealybugs, but you may only find a couple mealybugs within a cluster. So they can be kind of difficult to find. So I'd like to go through the life cycle of the grape mealybug because it's hard to manage something that you don't understand, right? So that's the first step in IPM, is to be able to understand and identify the pest and then understand the life cycle. So in the wintertime, So right now, probably those eggs are getting ready to hatch, but what we have in the vineyard are eggs that are under the bark. So the eggs are actually the diapausing stage over winter for this insect. And diapause is, if you think about it as it's a resting state. So it's hibernation. It's triggered by day length. So in the fall, as the days are starting to get shorter and the amount of light that we have in the day is shortening, that triggers the insect that winter is coming and it's time to enter its resting or hibernation state. So the state that does that for this particular insect is the egg stage. And then as we're getting closer to bud break, those eggs will hatch. So as the vine is, quote unquote, waking up, so are the insects. So those eggs hatch, and the nymphs that hatch out of those eggs are going to migrate up the vine into the spurs or onto the canes because eventually they're going to be out feeding on the leaves. And so getting closer up to the leaves as they're getting ready to do that. So that's the next stage in the life cycle is that after bud break, as soon as we have leaves on the vine, you're starting to see those nymph stages moving out onto the leaves and then feeding on the leaves. That's a really important time of the year because it's a potential time when we can make an intervention. So mealybugs, when they're under the bark, it's really difficult to contact them with insecticides. And so once they go out onto the leaves, it's much easier to kill them with contact insecticides. And so this time of year, like just after bud break, once they start moving onto the leaves, can be a really good time to make an intervention with an insecticide. Either a conventional insecticide or an organic product. And I'll talk a little bit more about the insecticide applications and timing of them in the later part of the talk. Okay, so those mealybugs have moved out onto the leaves, and then they mature, so they grow and mature, they become adults, either females or males, and mating happens. After the females are mated, they generally move back under the bark to lay eggs. So mealybugs are adapted to laying eggs in these sort of protected locations. That makes sense. Like, why would you lay your eggs way out in the public where a predator might come along and eat them? And so when they're ready to lay eggs, most of the females will move back under the bark to lay eggs. So that's happening sometime after bloom. And then in the summer, we have our second generation of nymphs. So those summer nymphs coming out from the bark again. And this time they're moving into the cluster. So it's close to veraison at that time of year. And so they start moving into the clusters. Makes sense if the vine is putting sugars into the clusters at that time and you're a phloem feeding insect, it's a logical place for you to go. So they'll spend their time in the clusters until that generation matures. Once that generation matures, which is usually before harvest or around harvest, we have mature females that move again back under the bark, and then those eggs will overwinter again, and the life cycle starts over again. So, as I mentioned, because this is a sporadic pest, the populations are also often small, and because it spends a part of its life cycle under the bark, it can be really difficult to detect it by scouting. So it takes a lot of time, it takes a lot of energy, and it's just not a very reliable way. Visual scouting is not a very reliable way to get a good understanding of what the populations are doing. It also means that you get like a lot of bark in your face, which is, I did that for a number of years and it's just not that fun. So one of the first things that was a real innovation for us is that we were able to use pheromone baited traps for monitoring. So now instead of going out and searching under the bark, searching in clusters or on leaves, we're able to use a synthetic version of the sex pheromone. So when the female is ready to mate, she produces a sex pheromone, which is what the male uses to orient to the female for mating. So this is the male stage here. It's the only stage of the life cycle that has wings. So only stage that can fly. And then when they get caught in the traps, that's what they look like. So basically, that sex pheromone, we identified the principal components of the pheromone. Those are synthetically manufactured in a laboratory, loaded into a lure. You place the lure into the trap, put the trap into the vineyard at the height of the cordon or the fruiting wire for 2 to 3 weeks. You bring the trap back into the lab to read it, look for males, and magically you have males caught in the traps, and so you can follow the phenology of this insect. So the next part of the talk, I'll talk about what we've learned about male flight data, and how we can use the information that we gain from those traps to make management decisions. So here are some flight data that were published from Washington, from some work that Brian Bader did while he was in Doug Walsh's lab in 2010 and 2011. And so you're looking at the month of the year across the x-axis, and then the number of male mealybugs in the trap on the y-axis. So following the phenology at the beginning of this growing season, we're not catching males in the traps. And the reason is because there are no males present. So at that time of the year, all the mealybugs are in the nymph life stage. And so we're not finding any males in our traps. Starting sometime late May, early June, we start to get an uptick. So that's the first flight. That's when males are flying, mating is happening, and then those mated females, as we discussed, go back under the bark to lay eggs. And then in that sort of July to August period, the trap numbers drop once again to 0. And again, that's because we have nymphs in the field, so no males in the field. And then once that next generation or that summer generation matures, then we start capturing males again, and we get our second flight. Mating happens again, and then the eggs go to overwinter. So the traps really allow us to look at the seasonal phenology within a single season, and then also to compare them between seasons. So you can see that in 2010, there was a much larger flight, and it also started a little bit earlier than it did in 2011. So the beauty of doing this every year is that you really get an understanding exactly of what's going on during that particular growing season. And there is a slight, it's somewhat consistent, but there are some slight fluctuations from year to year. So that's why we monitor on a yearly basis with these traps. Okay. And so with those traps, though, as I said, when you have low trap numbers, that's because there's no males present, because the nymph is the principal stage, or in the case of overwintering, the eggs are present. And many of the insecticides, particularly the contact insecticides, are most effective against the nymphs. And so by knowing that the traps are not capturing any males, that allows us to say, okay, there's nymphs in the field at this time, and to make our insecticide applications accordingly. Whether you do that with a conventional insecticide, the growth regulators, so that's buprofezin or Applaud, it can no longer be used in season, really, because of MRL issues, but it can still be used to lay dormant. So there's that time in the spring when you can, when the nymphs are on the leaves, and then Applaud could be used at that time. And then the systemic insecticides, so it takes a little bit of time between when those insecticides are applied, either to the leaves or taken up by the roots from a fertigation, or not fertigation, but through the drip, an application through the drip system. And so you're going to want to do those at the time of year, if it's going to take 2 to 4 weeks for that insecticide to be distributed throughout the vine, then that application can occur, around when you're having peak flight, so that by the time the nymphs are present, you actually have insecticides in the parts of the vine. For organic growers, we tend to use the soaps and oils. Those are the most effective products for organic growers. And they could be used either during that spring period or in the summer. The better timing, of course, is the spring, because in the summer generation, those nymphs are in the clusters. So you can do that somewhat, but then it can also be difficult to get penetration of those products into the clusters. We don't, as a sort of aside, we don't really use the pyrethroids for mealybugs. They're not particularly effective for mealybugs, and they're also quite disruptive to our beneficial insects. So part of the reason why we don't have large grape mealybug populations is because we have biological control, and so the pyrethroids can actually disrupt that and result in larger populations of grape mealybug than if you didn't spray at all. Okay. The other option for management is, of course, biological control. So I mentioned that there are parasitoids that will attack mealybug species, but they're not produced commercially by any insecticide, by any insectary. So you can't buy them, but what you can buy are mealybug destroyer beetles. And that's something that, particularly for organic growers, could be used during that summer period. And the reason why I say it could be used in the summer period and not in the spring has to do with the beetles themselves. So this is a tropical beetle species from sort of the tropical Pacific region. The female beetle, which is what you saw in this photograph, she will lay her eggs near the eggs of a mealybug. And then when the nymph of the beetle hatches, it basically just eats all of the eggs. So in this case, these were all places where there were either eggs or nymphs of mealybugs. And now all you can see left are the mealybug destroyer larvae. And over here in the video is just for you to see one somewhat live and how they move around on the vine. And the reason why we use them in the summer is because this is a tropical species. So if it's not almost 70 degrees, those females will not feed or lay eggs. And peak larva activity is around 82 degrees Fahrenheit. So they could be used in the spring if those weather conditions are met. But if those weather conditions are not met, then that's why we use them as a tool during the summertime. Okay. So that's an introduction to the biology of the mealybug itself. But the reason why we're here today is to talk about how the mealybug moves leaf roll virus 3, right? So that's transmission biology. So a mealybug nymph can acquire, within 1 hour of feeding on a vine that is infected with leaf roll 3, it can pick up that virus. And then if that mealybug moves to a clean vine or an uninfected vine, within 1 hour of feeding, it can transmit that virus to the healthy vine. So within 2 hours, you can have a vine, the virus move from one vine to another. Of course, maximum efficiency happens in around 24 hours, but it can happen quite quickly. The good news is that if the mealybug feeds for 4 days on a non-host of leaf roll, so if it keeps moving, keeps feeding on vines that are not infected, it loses the ability to transmit the virus. So it's not like once the mealybug picks up the virus, then it's infected for life. So that's a bit of good news, I think. And one of the most important things that we needed to learn for this project was about transmission and how it's happening in the field. And so in 2011, There I am out in the vineyard, and in this petri dish, I have mealybugs that were reared in the laboratory, and they were feeding on vines that were infected with leaf roll 3 in the laboratory. We've transported them out into the vineyard, and I am gingerly trying to pick them up without killing them, and place them on the leaves of these commercial vines. And the reason why we did this is because we wanted to understand the parameters of transmission in the field, because as we know, both insects and plants and pathogens behave very differently in a greenhouse than they do under commercial farming conditions. So somebody was nice enough to lend us some uh, nice and generous, and that they lent us healthy vines, which of course we infected when we're not able to cure them. So after we did the inoculations, every week I returned to the vineyard to look at those vines to determine whether they were developing symptoms or not. And what we found is that it took a year between the time when we inoculated the vines with mealybugs with leaf roll 3 and between when we started seeing symptoms. So I didn't see symptoms. I was checking every week until late summer 2012. And that was just a really important thing to know. So it's a 1-year incubation period between when the vine gets infected and when we can tell that it's infected because it has the visual symptoms of leaf roll 3. Of course, you can test these vines earlier than that, and some of them will test positive. They won't actually all really test positive until they've had 1 full year of symptom development. So. And other important aspects of transmission biology are that small nymphs are the most important stage for transmission. And there's 2 reasons for that. One reason is that they are the most mobile stages. So mealybugs may be a little bit like us. As they get older, they're less willing and more reluctant to move around. And so it's really the young stages that do most of the movement and most of the dispersal. They're also doing a lot more feeding. Why? Because they're growing, and also because if they're not feeding, when they're small, they have to stay hydrated. And so if they do not feed, they get dehydrated and die. So the young stages move the most, and they also feed the most. So they're the most important for transmission. The other important thing to note is that most movement of mealybugs is rather short distance. So when we see infected vines, the most likely vines to get infected next are the ones that are the nearest neighbors to those infected vines. So now let's talk about what it takes to manage leaf roll disease. So it requires technical knowledge. So we need to understand the mealybug, the vector, the plant, how it's reacting. There are, of course, economic considerations because everything that we do has an economic consideration. And then there's also all these social behavioral considerations. So how decision-making processes are carried out within an organization, whether somebody decides to manage leaf roll or not, and then what tools they're going to use to do the management. So the 3 main aspects of leaf roll disease management that are important are plant material, disease vine mapping and removal, and then vector detection and management. So I'll talk about each of those separately. From a plant material standpoint, ideally we would have plant material that is absolutely free from all pathogens and pests. I don't know that we achieve that, but we do our best, and we do that through certification programs that are the joint programs between regulators, a nursery industry, and then the growers or the people who are buying the plant material. And just as a side note, not only can leaf roll be moved on infected plants, but also vine mealybug in particular has moved around the state of California on infected plant material. So that's what you're seeing here, is that we had these green growing vines that had just been received by the buyer, and they are already infected with mealybugs underneath the wax that's covering the graft union. So we do have some resources on our website for hot water dips and then inspections for vine mealybug. Okay. So the next aspect I'll talk about is vector detection and management. The biggest challenges that we had when I started working with leaf roll disease in Napa is that there was a lot of uncertainty around mealybugs as a vector. So because people couldn't see mealybugs or they couldn't find them in their vineyards, they did not want to believe that they were the vectors that were responsible for moving the disease around. So there was insinuations that it could be phylloxera, that it was nematodes, that it had to be something that um, was not mealybugs. Well, I mean, people couldn't see nematodes either, but they were still convinced that that was what was moving them around. So we did some work, and mainly the pheromone traps were what was most important in reducing uncertainty. And then, as I said, we need to understand the life cycle, and we also needed to understand the seasonality. So even people who knew that they had mealybugs were really unclear about when they should be treating for them because they didn't understand the life cycle very well. Just to reinforce the point about how important it is to recognize mealybugs as a vector, we did some surveys of growers and looking at adoption of these 5 practices and the factors that are affecting adoption. So if you see adoption on the positive side, those are reasons why people were adopting. So people were adopting virus testing in the vineyard because they believe that there is important and they understand the importance of the controlling leaf roll disease. On the negative side, there's a couple of factors that lead to decreased adoption of these practices. One of those, of course, are economics. That makes sense. If it's too expensive to test for viruses or it's too expensive to manage mealybugs, then you're not going to do that. The other thing that I found was really important is that people who believe that mealybugs are not a vector or they don't think that they have any mealybugs in their vineyard are going to be less likely to adopt some of these really important strategies, and not just the vector related strategies, but all of the important strategies. So reducing this uncertainty and clarifying that mealybugs are truly vectors of leaf roll was a really critical aspect of our successful disease program. So I can say to you that you should be certain that mealybugs are vectors, and also that even if you're not seeing them, it's highly likely that you have them in your vineyard. So I won't belabor too much more because we already talked about the pheromone-baited traps, but just to say that anyone who doubted whether they had mealybugs or not, I would hand them a trap with a lure in it, tell them to go put it out in the vineyard and come back and see me 3 weeks later because they would probably have a lot of mealybugs in that trap. So that was a really effective way, not only for us to understand the life cycle, but also to get people to understand that mealybugs are present in vineyards. And even if we're not seeing them with our eye, they are a really important vector of leaf roll disease. So the other thing that pheromones, the identification of the sex pheromone allowed us to do is to develop mating disruption as a technology. So it's currently developed and commercially available for vine mealybug. And I understand that there is work going on in Washington, in fact, to look at mating disruption and how and whether it would be effective for grape mealybugs. Other things we learn is that the timing of the first flight is fairly consistent. So you saw this in the Washington data. We have a very sort of steep peak. And while the number of males that we catch may differ by year, there's a pretty good understanding of, in Napa anyway, that flight happens between, late April and late May. The second flight, not so clear, probably because of environmental factors, and then also just a little bit more stretching out of that generation. So when it's too hot, we find that we don't catch that many males in the vineyard. So if you had a heat spike, that can lead to some of the ups and downs that we see in that second flight. Uh, The other thing that trapping allowed us to do was to do some regional trapping. So through a public-private partnership and a grower self-assessment, we have in Napa what's called the Pest and Disease Control District. So those funds are collected, they're aggregated through the Ag Commissioner's office, and then a board of 5 growers decides how they're going to spend those funds. Since 2012, we've been doing detection trapping across the county for vine mealybug. These data are publicly available on the Ag Commissioner's website, and so people can access them and say, okay, within this area where I'm farming, are we detecting vine mealybug males? And that was a really important way for us to raise awareness, and then for people to understand how vine mealybug was moving throughout Napa County. And the other thing that we did with the advent of the pheromone beta traps is that for grape mealybug, we had neighborhood trapping programs. So basically, this was because neighbors were worried about whether leaf roll disease was spreading in their neighborhood, and they wanted to coordinate their actions. And so the neighbors were putting out traps, and then they were sharing that information. And so this is just a different way of looking at it, but you can see here the difference between years and the size of the flight. So the number of total males that were caught, and then also that peak in early May, and then looking at the second flight here in late August and early September. Another way to look at those data are to show them on a map. So these are maps that represent 2 different regional groups that were coordinating and trapping in Napa County from 2012 to 2015. And the dotted lines are the boundaries of the growers, and then the dots that you see are the traps. And the color and the shape of them shows how those traps are related to the neighboring traps. So when they're all the same color and the same shape across grower boundaries, that means that those traps are very similar to each other. So we saw these similarities of trap counts within regions. And then also sometimes an individual grower would sort of separate out from the region. And so what this told us is that there were regional patterns of grape mealybug populations. And so sometimes an individual action could result in a difference for that region. And then other times the populations of grape mealybug were influenced across a region, across various different growers. So it's perhaps environmental factors that were affecting that. As Michelle mentioned in the introduction, I was in Washington last January, and so we were trying to get people in that area to get excited about sharing and grape mealybug trap data. And so this is a screenshot of the sharing the monitoring that was done last season, and then how we were sharing that so that all growers could have access not only to their own data, but then also to look at how their data compared to other growers within the different regions that we were targeting. So we could again look at individual trends in grape mealybug population, male populations, but then also look at regional trends, so regional and individual trends. And Michelle's going to talk a little bit more about that information, what we learned from it, and then provide some resources and talk about where that effort might be going for you guys into the future. Okay. So leaving the vector aspect behind for the rest of the presentation, I'm going to talk about disease vine mapping and removal. And again, I'll talk about some of the challenges that we had when we were starting this leaf roll program in Napa. Roguing was perceived as ineffective, and leaf roll was persisting. So people were frustrated because they felt like they were removing disease vines, but again, it was persisting. And then, of course, we had economic considerations, and we wanted to understand at what densities of disease incidents should we make different management decisions. So when we think about roguing, a couple things were really important to helping growers in Napa understand the importance of roguing. One of those was spying on neighbors. And in this case, these neighbors were in South Africa and also in New Zealand. What we were seeing is that growers and regions in South Africa and New Zealand who were removing vines were having success stories. And so they were sharing that information with us here, mainly because people like Gerhard and Von Bell were coming to California and talking about the successes that they were having. So that knowledge sharing is really important, which is probably why I've been invited to talk to you about some of the great results that we've had in Napa and giving you confidence that you could have those same results in Washington. The second aspect that was really important to really emphasize roguing is, as I already talked about, that field trial. So when people were frustrated because they felt like the disease was persisting despite the fact that they had removed vines, we were able to now tell them that it takes a year between when they become infected and when they show symptoms. So you're always a little bit behind on your roguing efforts. But if you have a sustained program where you're roguing every year, you can catch up and you can keep levels below 1% disease incidence. The other thing we were doing is that in our regional groups, we had early adopters. So people who were convinced either because they heard it from someone else or they had the information on the roguing studies that it was successful, and so they started roguing, and in our regional groups they were telling their neighbors, yes, roguing works. Here we did it in our blocks and we're having success, and now we don't have that much leaf roll disease. So those are really the three aspects that were really most important to change the perceptions that we had locally about roguing and about its importance. So I did mention that it needs to be a multi-year strategy because you're that 1 year behind. You're slowly reducing the amount of disease incidence over time if you're just removing the symptomatic vines. So it can take between 3 and 5 years to really get to levels where you're having less than or 1 or 5% disease incidence every year. Ideally, we're aiming for that kind of less than 1% every year, and we are able to achieve that with multi-year roguing strategies. Of course, one of the obstacles to successfully removing vines is being able to identify the visual symptoms of leaf roll disease. So we did spend a lot of time in the early days just thinking about, okay, what does leaf roll look like compared to, say, spider mite damage or compared to red blotch disease. In some of our white cultivars like Chardonnay, the leaves do roll, so we can see them even if the leaves don't turn red. But then we also have asymptomatic cultivars. So Sauvignon Blanc does not show any symptoms that we're aware of for either leaf roll or red blotch. And so removing those vines can be really challenging because it has to be done by testing, and it's really expensive. So when you're starting a visual symptom mapping program, there's a couple of things that you want to consider. It can take a while to learn and be really practiced and good at distinguishing leaf roll from other reasons why the leaves turn red. And so when you're starting that, you want to make sure that you're investing and you're giving your staff who are doing that, whether they're interns, whether they're viticulturists, whether they're members of your crew, that you're giving them enough training and mentoring and allowing them to take a bit of time before they become proficient. So in my experience, it can take at least a full season of mapping, and sometimes, and some people, it takes them 2 to 3 years to really become proficient, where they're really confident and then they can walk out to a block and say that, yes, that's leaf roll, or that's red blotch, or that's potassium deficiency. So you want to really invest in training those staff. And then also, as you can, try to retain them, because once they are proficient, they can recognize that. It's a big investment, and it's nice to keep them as long as you can. And then lastly, you want to be consistent in your mapping. So if you do map, do it on have practices that you follow. And if you bother to map them, please go back and remove them, because otherwise, why put in all the effort of mapping? And then when you think about your replant strategy, how are you going to, reestablish vines in some of those places? And that can also require a lot of effort. So we have worked on some decision support tools that can help with visual symptom identification. One of the tools that we worked on early on was airborne hyperspectral imaging. That's based on the fact that an infected leaf will have a different a spectrum of light that's reflected than a healthy leaf. And when you have a sensor, it can pick up those differences, and then you can have a computer model that then will then tell you, okay, yes, that leaf is infected, so that vine is infected. And we had fairly high levels of confidence for Leafroll with airborne imaging, both looking at agreement of when we had infected vines and then not picking up vines. So no false positives. So vines that were not infected were not being marked either by our scouts or by the cameras being infected. So of course it's not perfect, but we're not perfect as humans either. So I don't know why we would expect a machine to be 100% perfect either. And then just the last thing to say on that is that in Napa anyway, there is a company that offers that as a service. I'm not sure whether that's something that is also extended into Washington, but it could be something that you could explore. Excuse me. Because the hyperspectral imaging worked really well for Leafroll, but not so well for red blotch, we're also looking at a machine learning. So basically that means that we've collected 4,675 photographs of either red blotch, leaf roll, abiotic girdling, spider mites, or healthy vines. We fed those images into an algorithm and in the computer, and then that model is then loaded into an application that we're calling Virus Vision. So you load the app on your phone, you start the imaging from the home screen, and you do continuous imaging collection for about 10 to 15 seconds. After that, when you hit stop, you get a confidence rating. So the app will tell you how it quote unquote diagnosed that that vine, and then the confidence level that it has. So right now, because of the database of images that we have, we're only rating in 4 categories. So leaf roll, red blotch, other, which means that it's red, but we don't think the reddening is caused by either leaf roll or red blotch, or asymptomatic, meaning that it doesn't have any symptoms. And as we get more photographs, we could potentially take that other category and divide it out into, say, okay, that's maybe spider mites, that's maybe potassium deficiency, that's some kind of physical damage or girdling. But for now, we've just lumped them all into other. So in 2023, we tested this. We tested it with over 1,000 readings, and looked at how the results that we got from the Virus Vision compared to our visual assessment. So we had really high agreement, almost 97% for red blotch and about 92% for the non-symptomatic vines. And the reason why we had such high agreement for those is that we have a lot of vines in those categories. Our assessments were slightly less with the leaf roll disease vines, and that's simply because when we started collecting images, we didn't have that much leaf roll left in Napa anymore. So we actually don't have that as many images as we need for the app to be as confident in a leaf roll assessment. So we've been gathering some more images, and we'll need to update the model, which would hopefully improve our leaf roll assessments. We are still working on this with Thule, which is now a part of CropX. And so if you're interested in becoming a beta user, you can reach out to Sarah McDonald on my team at that email address, and we can get you added. And then you can test this in Washington in your vineyards and see how it's working. Okay. So the last critical thing to think about when we're talking about disease management is when should we implement each practice? So what different levels of disease incidence sort of trigger differences in our management plan? Economic studies have attempted to answer this question through modeling. So basically they've set about a 25% disease incidence threshold. Below that threshold, suggesting that the most economical approach is to rogue and replant infected vines individually. And above that 25% threshold, the suggestion is that it's most economical to redevelop the entire block. Those are great guidelines. Maybe they work for you. In our case, there's just a lot of diversity of, farming philosophies across Napa Valley. And so what we found is that both with our field studies and in our modeling, that it can be slightly more nuanced than that and context dependent. So for instance, in the leaf roll days, I was working in a heritage block. It had been planted more than 50 years ago, and the grower that was really important to the grower. It was really important to the fruit contract. And so they were reluctant to redevelop the entire block, even though they were above the 25% disease incidence threshold, because that block was so valuable to the wine program. So there's definitely some individual nuance that can go into that. And so let's talk about some of the field studies and modeling that go into decision making. So there's some nice work that came out of New Zealand by Vaughan Bell. He looked at 5 different disease incidence categories, 3 different mealybug abundances across those disease incidence categories. And he did that in both field studies. So he had actual data, and then he also did some modeling based on the data from the field studies. So it's a combination of field studies and modeling. And what he found is that regardless of initial disease incidence, roguing is the optimal response. And at low levels of mealybugs, roguing is still the optimal response. When you start to get to higher, moderate mealybug abundance, roguing becomes more expensive. So it's still the optimal response, but combining it with strategies that similarly are reducing mealybug or vector incidence leads for more success over a shorter period of time. So you always want to be roguing, but when you have moderate to high mealybug abundance, you also may need to be making some kind of intervention for mealybugs in order to sort of speed up the process and make it less expensive over the long term. We saw something similar in our work that we conducted in field trials from 2009 to 2016. So what came out of our work is that at less than 1% leaf roll disease incidence, the pathogen is probably originating outside the block. And so you want to rogue because you don't want the pathogen to build up in that block. But there's really no clear role for insecticides in that block simply because there's just not enough inoculum to really propel the disease forward based on how many mealybugs are in the block. So definitely be rogaine, but no clear role for insecticides in that block. And then because the pathogen is originating outside the block, that's when regional monitoring and communication between neighbors is really important. When you start getting to the 1 to 20% disease incidence, there's a role of both rogaine and insecticides. So in some cases, it was a combination. In other cases, it was one or the other. And at greater than 20% leaf roll disease incidence, rogaine is definitely necessary because you need to reduce the amount of inoculum, and insecticides did not reduce spread when they were used in that block. And the reason why they didn't is because there was so much inoculum. So in the less than 1% disease incidence, we don't use insecticides for grape mealybug because there's not enough inoculum. And in the 20%, we didn't see an impact of them because there was too much inoculum. So I will say, however, though, if that greater than 20% disease incidence is your source block for your less than 1% disease incidence, then perhaps there would be a role for using insecticides just to protect your other blocks, but not to reduce the amount of spread in that block that already has the greater than 20% disease incidence. Some work that came out of New York on spatial rogaine looked at removing not only the vine that had symptoms, but also 2 enroll neighbors on each side of that vine. So this work didn't come out until after we were basically done with a lot of our work in Napa. So I don't know how it might have sped up our process of getting leaf roll out of the vineyard. But what I can say is that this is a map of a block from Napa, and that all of these vines that are in pink were symptomatic and they were removed. And then it left these holes where there was just one vine in the middle of a sort of a vine desert. And so removing that vine along with those other vines probably would have made sense at the time. And also there was a great likelihood that that vine was going to end up infected or symptomatic within 1 to 3 years. So you would end up removing it anyway. So there is value in that spatial rogaine approach, particularly in areas where you have a high aggregation of leaf roll, because the risk that those vines are infected is pretty high. And then it just means that from a management perspective, you don't have a single vine out there that you have to, spray and then not spray anything, in where the vine spaces are. So just from a management approach, it's easier anyway. So we did think that even though we didn't use it at that time, that a spatial rogaine approach can be particularly useful in high incidence areas or areas where there's a lot of aggregation. Okay, so in summary, I'd say that when we started in 2009 working on leaf roll disease, there was a lot of uncertainty around leaf roll disease management. So not only did people question whether mealybugs were vectors, but they also just didn't really know which were the best steps that they could take to move forward. And so that uncertainty just made management more complicated. The other thing that was a problem at that time is that leaf roll was not universally recognized as a problem. So some people were saying it was fine, it wasn't a big deal, it may be an asset to wine quality, which is not true. And there was no sort of widespread commitment to management. And so what we did from 2012 to 2016 is that in addition to conducting these research trials, we were also working. So we were working at the level of the entire county, and then we were also working individually or in neighborhood groups. So there was a lot of peer networking going on, a lot of information sharing. As people were successful, they were sharing their successes, they were sharing their challenges as well. As I mentioned, we did widespread trapping for grape mealybug in these regional groups. And as we met monthly, we would share that information so that people can understand not only what populations we were seeing, but at what life stage they were in. So we were looking at the phenology through trapping. We did determine and we communicated very rigorously that roguing is the optimal response. Because this is an incurable disease, the only way to reduce inoculum is to take those vines out of production. Vector management for large populations is important. For grape mealybug, of course, if you have vine mealybug, it is a direct pest on its own, and so you're going to need to manage that. Luckily for you guys, you haven't had vine mealybug move into Washington yet. And we were doing some interventions in different cases, either with insecticides or predators for grape mealybug. And so what we have now is that as of 2024, we have very little leaf roll left in Napa. In fact, finding blocks that have leaf roll so I could take photos was really challenging. And unfortunately, we have a new challenge, which is red blotch disease, but that was interesting when we did our interview studies that people said that they would rather have leaf roll than red blotch, because at least now they know how to manage leaf roll and they know what the effective strategies are and when to implement them. So I'm hoping for similar success for all of you in Washington. So thank you to the project team who was involved in this work, and then, of course, all of our funding sources. And with that, I will close out and take questions.
Thank you, Monica. Do you mind if I quickly share some of our mealybug stuff from our last workshop with the team here? So as Monica mentioned, well, first, like Monica, let's maybe like really pound it as she leaves the screen. I think one thing that we in Washington took home from the last time we had Monica talk with us, and I think you emphasized really well today, is that the first and foremost strategy for leaf roll management is roguing, right? Roguing,
Yes.
right. And then the roguing plus mealybug management, or in this case, insecticide use, only it works really well when you're kind of at that middle ground where, you know, red blotch or not red blotch, leaf roll has been established in the vineyard and it's starting to spread a little bit more. But at no point is mealybug management only an effective way to stop the spread of leaf roll within a block or within a region. So I think we really need to kind of embrace that we're going to talk a lot about mealybugs and mealybug monitoring. Yes, lots of warm hugs here, but ultimately it is roguing. It has to start with roguing, because as we learned, as you showed, 1 hour of taking up the virus, 1 hour to spread. So if you have the virus there, you got to get rid of the virus first before you can manage, before insect management is important. So Monica had talked a lot about mealybug trapping when she was up here last time, and we put together a nice little article in our spring 2023 issue of Viticulture Ecology Extension News. And so I'm putting that in the chat box for folks right now. And that article talks about how you can get your own traps for mealybug monitoring. And it talks about where to buy the Delta traps, what kind of lures, what kind of sticky tape works well, frequency, timing of setting them up. But I think really important for folks up here in the Pacific Northwest, particularly Washington, is it's really important to get the right pheromone lure. Right. So because California is really concerned about vine mealybug, a lot of the traps that you could potentially find for mealybug are going to be vine mealybug pheromone lures. And grape mealybug is not attracted to a vine mealybug pheromone, just like vine mealybug is not attracted to the grape mealybug pheromone. So you could put up a lot of Delta traps, and if they are baited with the vine mealybug lure, you will have a lot of zero catches. So but you might feel great, and maybe you need that, but like it's not good. So make sure you are getting the right pheromone, and that's for grape mealybug here in Washington State. So grape mealybug is what we need the pheromone for. The other thing is Monica shared. I'm going to share my screen here real quick. It's probably not going to be very easy to see just because of colors, but Monica shared this mealybug monitoring pilot program we did last year, and her team down at UC and UC Napa was really, really great about setting this up for us. So they did all the work. And then we had some beta growers up here that sent weekly trap counts to me. And it was a lot of fun. We had a good time doing that. And so here you see folks down in Walla Walla. Traps got out a little bit late, but we caught the tail end of the first flight and we caught the second flight out here in August. The other great team was on Red Mountain. They started trapping early, so we have really nice first flight counts and then end of August. But I want to emphasize both from Brian Bader's work in 2010, 2011, which people remember was a really cool, there were two really cool years back to back relative to last year, which was pretty on average. The peaks are within about 2 weeks of each other across regions. So we like Monica saw in Napa, is most of the time we're going to be seeing our peak male mealybug flights towards that middle of May to middle of June. And we're going to see our second peak towards end of July through August, right? So this is great. What I loved about this particular data is it suggested to us that, one, maybe allowing everybody to participate who are monitoring mealybugs to import their data and we can see it across the region might be an excellent way for us to kind of think regionally when flight patterns are happening. Or two, maybe we can set up sentinel traps in certain key regions to help monitor. In particular, when Monica showed her nice little map of how mealybug populations were relatively either similar across regions or by grower management strategies, Doug Walsh's and part of Brian Bader's study in that showed that really one trap per 30 acres was pretty effective for kind of determining the population. And that was his minimum trap density. So he could have done 1 in 100. And maybe that works too. They just didn't monitor for that. That's probably a little bit light. But if you have a 30-acre block, you don't need to set up 20 traps to figure out what you actually might be getting to the point of pheromone mating disruption at that point, right? Monica and I've talked a little bit about that, but that could be very helpful for or even if you're in a smaller growing area, or if you have surrounding neighbors, is maybe go all in and you guys can share the data within that particular region rather than everybody individually trapping. What Monica allured to that we might be continuing this work is Gwen Hoheisel and I have put in a request through the Washington State Grape and Wine Research Program to fund the development of a grape decision aid system. And so for those of you who are not familiar with that, it's a very commonly used system in tree fruit here in Washington. I'm going to put a link to the tree fruit decision aid system here in the chat box. But what we are planning to do with that, it's a platform, right? So it costs money to build and launch. But once that's launched, it will tie our spray recommendations in with a whole bunch of stuff, timing, phenology models, et cetera. And perhaps one day we will either run a mealybug development model on that website or partner it with your user's ability to log in and enter your own trap data and have an onboard monitoring program, which then can feed information into the model, and using AI can every year improve the model's ability to predict when key times are happening. And once we know when key flight patterns or things like that are happening, we can then predict when you should be doing intervention or when you should be placing traps or when you should, as Monica showed, when you should be different times for systemics or contact insecticides. So all really cool stuff about monitoring and managing the mealybug, but step one is to rogue infected plants. I think at this time we can take any questions, so you guys can chat. If you can't use if the chat box has not been enabled, please use the Q&A function for Monica, and we can go from there. I'm going to stop sharing my screen here. So now's the time. Well, as folks are waiting, Monica, question for you. The thing we always get asked is, in the roguing process, how long do you have to wait, or should you wait, before you can replant those particular areas?
Yeah, it's a good question. If you have a lot of disease and it's a hot spot, it might be worthwhile to wait, a year or until, you're really on a downward trajectory. But if it's just, some individual ones, or, if it's just 1% or a very low percent, those vines can go, back in pretty quickly afterwards. So
kind of monitor, like if you have a lot of disease, wait until you and that makes economic sense, right? Wait till you can kind of plant all the young vines at the same time, so you're not managing different ages.
Yeah. Mm-hmm.
So Gareth asked how far an adult male mealybug could fly.
They're not great flyers, so they don't live very long as adults. It's pretty short lifespan as adults, and they get blown around, but they don't, yeah, they don't fly
Mhm.
particularly far. So,
You know,
but they can get picked up and blown in an air current. So they can disperse far, but they are not particularly strong flyers on their own. And of course, the males don't have mouth parts. So at that point in the life cycle, they are not moving the virus at all.
Mhm.
Their only job is to live long enough and move to mate with females at that point of their life cycle. And again, that part of their life cycle lasts 1 to 3 days, maybe.
Excellent.
Yeah.
So Monica, Ula asks, do you recommend spraying Movento? And if so, when?
So yeah, for mealybugs, Movento is probably the most effective product, particularly for vine mealybug, because the nice thing about Movento is that it will kill mealybugs that are under the bark.
Mhm.
And it falls into that category of what I was talking about of it is a systemic product, and it takes a little bit of time between when it's applied and when it is converted to the active form, and then um, translocated throughout the vine. So you're gonna want, if you're for great mealybug, you'll be targeting that. And also you can't spray it early season because you need a canopy to activate it, right? So we don't use Movento against that first generation of nymphs in the spring. What we use Movento for is for the summer generation. And so usually we're applying it around peak of the first flight, so that by the time those nymphs are hatching, the Movento is, active in the vine. So you go back, you need to dial back 3 to 4 weeks to make sure it's fully active. So that is around like when we're starting to get peak of that first flight.
And I think that's a good point there, Monica. Is like if you're going to use a systemic product like Movento, it is super important that you're pairing that with monitoring data or using those delta traps so you know when to apply. Because if you apply Movento a little too early or a little too late, you're going to miss that key window to have it be metabolized and available for when nymphs are around.
Yeah.
So step one, get some delta traps to start making sure that those are placed out by early April. Monitor them, and at the peak, you'll start to notice when trap numbers start to go up. That's when you can apply Movento for that to control the nymphs in that midsummer time period.
Yeah. And the nice thing is that usually that peak is happening at the same time that you have enough canopy. So, that those two things are overlapping.
Right. So actually, maybe we can share our dashboard here again. And so because we have that great data set from Red Mountain. So in that case, they had started trapping in May. And so if you were to use Red in this, and this is for last year, of course, right? So last year had they started to see that, trap numbers are starting, and it doesn't last. The counts just don't go for weeks and weeks and weeks on end. So they're pretty short. So once you start to see a rapid uptick is when you can apply a product like Movento, and then it'll be ready and available by the time the nymphs are out. So something to think about. Or when you start to see that first thing or first dip in peak flights. But when we start to see lots of numbers, and you're probably there.
You're getting there, yeah.
Exactly.
Yeah. And that's for grape mealybug only, right? So yeah. I'm not talking about vine mealybug at all today.
No vine today, just grape. Just grape. All right, so we'll give folks a little bit longer if they have any more questions we can put those right into the Q&A. Or Monica, I know many folks might be interested in providing you some leaf roll pictures to participate in your visual vine, or was it visual virus visual?
Yeah, virus vision, yeah.
Virus vision program. So I don't know if you have Sarah's email address you can type in the chat box for folks to see that.
Oh, yeah. Yeah, if they want to become, I don't think we're really collecting any more images, but if they want to participate as a beta user for the app, you can contact Sarah.
So we'll also ask if we can control grape mealybug through trapping. And if you want, yeah, so the answer is no on that. And so Ula, that's kind of the idea behind some of the work that Monica did with vine mealybug on mating disruption, and what Doug Walsh is doing up here with grape mealybug on mating disruption is essentially the pheromone that is used for mating disruption and the pheromone in traps are usually the same. It's the density at which you put them out, right? And so traps, you're not really going to be able to place enough out there to confuse the males so much that they die before they can mate, right? For an example, like we say, oh, place one delta pheromone trap, monitoring trap every 30 acres. With the baits, at least that I know Doug has looked at, it's like 50 lures per acre, right? So it's 50 per acre. So you can multiply that by 30 to give you the number that you would need over 30. So it's a much, much, much higher density. That's just not practical with a delta trap. Would you agree?
Yeah.
And then Gareth wants to know, is leaf roll where in the plant is the leaf roll virus present? Can it be in the roots?
Yes, it can be in the roots. But the caveat is that even if the virus is in the roots, that doesn't mean that it can be transmitted from there. So grape mealybugs do not go onto the roots. So there's no way for them to pick up virus from the roots. And grapevines don't root graft, so you can't have transmission, vine to vine transmission through roots either. So even though the pathogen is in the roots, it's not epidemiologically significant, because there's no way that the vector can move it from those roots to another vine. And even vine mealybug is only found feeding on the roots in the really hottest parts of California, so down in the Coachella Valley. And the reason is just because it goes onto the roots because it's so hot. But even in other parts of California, we don't see vine mealybug on the roots. So mealybugs they're not adapted to live in a root ecosystem. So.
Now we do have some growers that get nervous about when they're doing whole block removal, right? And you have root tissue and crown tissue, and then that does, because we have vines that then sprout or redevelop from that. So yes, that new tissue could be a carrier for leaf roll, and if mealybugs go on that and you don't remove that tissue prior to planting the new vines, and that could be a source. But that's not from root grafting; that's from exposed foliage tissue.
Exactly. Yeah, that's the only way that could be a problem. But yeah, then your follow-up question, you don't need, as long as whatever's left out there is not going to grow again, you don't need to do any soil applications or anything after roking.
If you're really I think we did a paper or a project with a grower here in Washington where we were trying to, how do you best really kill a plant, a leaf roll infected vine, right? And there's a lot of crazy ways you can do that. But in the end, if you don't get rid of any surviving tissue, then there's still potential presence for virus. But most typical roguing practices, if you're doing a good job at removing the vine, that's step one. And then all the other stuff in between are just bedding cleanup, if you will.
Yeah. Don't just cut off the trunk and, leave it out there to resprout, right?
All right. So we'll leave like one more minute for questions in case there are any. And Monica has placed Sarah's email address in the chat box for those that are interested in that, to reach out to Sarah to learn more about the visual ID. Because Monica, we have enough leaf roll pictures that we can send you to help that machine learning in its learning processes.
That'd be great. And if you guys are doing some testing of it, it'd be great to see it tested outside California, and then also just, with leaf roll under your, canopy and sunlight conditions. So we can, if we get a lot of people who are interested from Washington, we can set up a webinar kind of later in the season where we can just talk through, how you use the app, and then you guys could feed us data and tell us how it's doing too. So that'd be great.
Perfect. And so, Monica, you did talk about it earlier in your presentation on how mating disruption has worked, or some efforts, but could you do a quick refresher on how mating disruption has worked, or some of the caveats to it? And I think you guys mostly have used it for vine mealybug attempts, correct?
Yeah, so I haven't actually seen any of the information on how it's working for grape mealybug, so you might know more about that than I do. But I know for vine mealybug, it's really effective, not only to reduce mealybug populations, but also when we use mating disruption for vine mealybug, it really reduces virus spread too. So the two in combination are really important. With mating disruption, there's a couple important things that you need to do. You need to use it consistently. So if you're in a mating disruption program, don't use it one year, then not the next year, because it works by over time, reducing mating, because mating still happens, and so you're reducing a population slowly over time. So what we see is that after 3 years of using mating disruption for vine mealybug, then we have, this really nice reduction. So once you invest in it as a program, you really should just, continue to use it for a continuous period. Regional programs work really well because with mating disruption, the larger area just provides it really reduces spread at a regional level. So because it reduces those hot spots, and what else do I have to say about it? That's probably about it. But
Yes.
it's really important for us for vine mealybug.
Right. And so Doug Walsh is currently working on some of that work up here for grape mealybug. He just presented on some of that work actually last week, and it's looking really good. The fundamental difference is the pheromone for grape mealybug is much more difficult to synthesize and much more expensive than for vine mealybug. And so because of that, and because leaf roll and grape mealybug are not as big of a concern in California, which is where most of the grape acreage is, and in vine mealybug is a concern, a lot of the commercial companies have focused on that, right? And so grape mealybug pheromone production has been very secondary for a lot of companies. And so Doug is doing some excellent work on how many lures and how long they last. And some of them last actually 2 years. They're still exuding pheromones. So they're really great potential there. It's just a matter of they're not producing enough of it to really be commercially viable right now. But I see that, it's kind of, if you build it, they will come kind of thing. So I think once more and more people request, maybe some of that will increase. So
maybe it will. I mean, definitely while we were in the midst of our leaf roll program, people were asking for mating disruption for grape mealybug in California, and we couldn't get any companies to even bite at all. So the fact that somebody's even making or testing some now, I think is a big step. So hopefully that will be.
We're waiting. And I think there's been some recent discoveries in how to speed up the synthesis for the pheromone. So that's a biology component that I think was a limiting factor. And so Gareth wants to know if virus, especially leaf roll, can be spread via things like pruning tools.
No.
Yeah.
So it's a very specific interaction between being picked up by a phloem feeding insect and then moved in the phloem to another vine. So it's an RNA virus. It's not that stable outside of the plant or outside of the insect. So it really has to be moved by an insect. Aside from, of course, if you graft material on, so grafting, of course, is one way that you can move it physically, but grafting is the only way physically that it can be moved.
Absolutely. It's also the reason why if you are ever in the process of replanting a vineyard, we do recommend you don't necessarily just take cuttings straight from your own block to propagate, especially if you're having problems. Start with clean plants, right, Monica? Step one, clean plants. Step two, rogue when things are infected. Then step three, think about insect control.
Exactly.
All right. So we do have a couple minutes before our 4 o'clock hour. We can go to 4 if we have any more questions, but we'll leave it open for just a little bit longer, and then we will thank Monica for participating, because it's so fantastic to listen to your talk every time. You learn something new. Oh, I learn something new every time. It's great.
That's good.
And we're happy you could take some time off in your afternoon for us on Leap Day, on the day that software apparently doesn't work sometimes because it doesn't recognize the date.
Well,
Hey,
thanks.
so Michelle, this is Jen
Yes.
here with
Hi,
the
Jen.
Washington Wine Industry Foundation. And as an organization, we just want to thank both Michelle and Monica for their time and effort today, especially with their patience when it came to getting this up and running when Zoom doesn't realize that February 29th is actually a date. So, but yes, Michelle, I have to say I learn something every time I listen to this as well. And I'm not a grower, but I find it very interesting. Monica, thank you for all of your expertise, and Michelle for all of your coordination, and for all the attendees, thank you for joining us today.
Absolutely. Thank you, guys. And Monica, again, thanks for giving us a little silver lining that we can potentially move forward with our Leafroll changes. So thank you.
Definitely can. Thank you. So
Absolutely.
good afternoon, everyone.
All right. Thank you, everybody. Thanks for attending.